Looks like people are joining. Okay. I think I can start making my opening remarks. So I would like to send greetings to everyone who is participating in our first EMX seminar of 2021. I would like to say Happy New Year to everyone, but that doesn't seem quite appropriate. But let's hope for a better new year than the one which we passed. You know, science brings us together across borders and cultures. And TEM, as a subset of science, also brings us together across disciplines, from biology to physics, engineering to medicine. And so this multidisciplinary format of this symposium, this seminar series, is meant to encourage us to recognize the different advances which are being made and have been made across electron microscopy as applied in important aspects of science. And with this spirit in mind, we have two great speakers again, and I'd like to thank them for being totally good sports to get us going so early in the new year, Joachim Frank and Jennifer Dionne. And I won't delay matters any further. I'll pass the baton over to Professor Hua Chu to introduce Joachim.
Thank you, Bob. Happy New Year to everyone. And thank you for joining our first Stanford Electron Microscopy X Symposium series of this year. It's my great privilege to introduce today's speakers, Professor Joachim Frank from Columbia University in New York. Joachim is the 2017 Nobel laureate in chemistry, which he shared with Richard Henderson and Jacques Dubussy. Throughout the years, Joachim has developed many fundamental and practical approaches to determine 3D molecular structures of biological molecules using single particle images recorded in a transmission electron microscope. Joachim is truly the one who have made single particle cryo-electron microscopy possible since its beginning. Thank you, Joachim, for your leadership and innovations. Joachim is also the first investigator in our field to adopt an open source software policy. His generous sharing of his single particle reconstruction software called SPIDER has benefited and inspired many early practitioners of single particle cryo-EM, including myself. Joachim today is going to deliver his lecture, how he can now determine not only the static, but also the dynamic structure of cellular machine. Joachim, thank you for joining us today. Please take the floor.
Okay. Thank you very much for the invitation, which is truly the first one for this year. And I hope that the year will be a better one than last year, which I guess sets a very low bar. So I'm going to share the screen. You see the screen? Hello?
Yes,
yep, it's great.
Yeah, you need to put it in the, yeah.
Okay, so Happy New Year. This is, sorry, this is my title right now, Capturing Motion by Cryo-EM. And here we are in cryo. This is this morning's picture and I wish you all a happy new year. I'm here in Great Barrington, Massachusetts. So I'm going to start by talking about molecular machines in the cell. Many molecules act in concert in a processive way. And we wish to know the structures of all components, but also the way they interact dynamically. And there's an important principle of reductionism that we use in approaching structure by single particle methods. We study a subsystem in isolation in vitro, and we hope that this somehow approximates the processes in the environment of the cell. Of course, there are other techniques, especially electron tomography, which is going to take over in the future and hopefully gets us in to similar resolution ranges. But right now, single particle cryo-EM is the most powerful technique to look at both static structures and dynamic structures. And I'm showing that this brought out because he's known for a paper that he published in the Cell, and he points out that we have vastly what's going on in the cell, that it actually works not as a sort of a random aggregate of molecules. So here are examples of molecular machines: ATP synthase, RNA polymerase, with DNA into mRNA. Ribosome is my favorite because I've been studying it for a long time. It makes proteins, chaperone folds proteins, dynein transport molecules, spliceosome edits mRNA, flagellum motor, and many, many others. So first of all, I have to tell you about principle for observing motion by single particle cryo-EM. Observing motion by something that freezes is something of a paradox, and we can only go by snapshots. Now, standard cryo-EM gives us snapshots either of an unchanged molecule, if the molecule in fact doesn't undergo any changes, or it gives us snapshots of conformational changes that happen all the time in equilibrium, in the thermal equilibrium. And an example here is the ribosome that is found in two conformations, ratchet-like formation. The other one is another kind of investigation which cannot be done by standard cryo-EM, which is capturing short-lived states in a reaction of two or more components of a machine on the way toward the equilibrium. So we don't have equilibrium. We put components. Is too fast to be observed by standard cryo-EM because it takes a few seconds at least to do the necessary manual labor. And so the latter part, it can be done with time-resolved cryo-EM. And here I show two components of a reaction, and then there's an intermediate, and then when we wait as long as maybe 600 milliseconds, then we get the final state. So in between, this is what we try to address. Now, the ordinary cryo-EM, you have already heard a lecture by Richard Henderson, if not lectures that you have heard before. pipetting, blotting, blunt freezing, and here is a commercial instrument, and this is here the principle. The grid, a little droplet is put on the grid, and excess is blotted away, and then the grid is very quickly immersed into the cryogen. And these are the now the big machines that sold all over a very wonderful way. So here I'm just giving you an idea of what is happening in standard cryo-EM and how the snapshots give us an idea of motion. Okay. Okay, so we're looking at a copper grid at increasing magnifications right there. Okay, so we have these holes that perforate the carbon, and carbon is top of the grid. And now we look at this here. Okay, so here we see all the molecules. And the molecules actually move between different conformational states in their thermal equilibrium. And then this is in solution, and then it's quickly immersed into the electron microscope. You we see this purple and because of the high amount of noise. And then we have tools that detect the positions of the molecules and extract some kind of a sorting classification algorithm that the different data sets apart. So then we get reconstruction from each class. So we, in a nutshell, we can say we take microclass of molecules, we get some sophisticated data processing, and then we get near atomic 3D map or several 3D maps. So that's sort of what a lot of people are doing, including the people at Stanford. Now I'm coming into the Timeresolved techniques. They were introduced by Berriman and Arnwin in 1994. And the idea is that you have a particular molecule already on the grid, and it's prepared in the standard way by pipetting and blotting. And then as the EM grid falls down into the cryogen, it is then sprayed by the other component. In this case, we have the acetylcholine receptor here on the specimen grid, and it's sprayed with acetylcholine. And in the process, the channel changes its structure, and this can then be recorded here by standard EM investigation and processing. But I'm calling this spraying mixing because the mixing actually occurs on the grid itself. Okay, one problem with this is that the mixing mixing and reaction cannot be controlled very well. So one has all kinds of reactions, parts of the molecule that don't see the second component. Now, mixing spraying is another idea in which the mixing is done in a controlled environment, and then the reaction is also done in a controlled environment. And then the resulting reaction product is sprayed on the grid. So we're talking about short lives in the range of 10 to 1,000 milliseconds. We are using the principle of microfluidic chip that was developed in a collaboration between RPI and Wadsworth at Albany back in 2000, in the 2000s. And now we adopted this whole technology at Columbia, And we are using a new organic polymer, PDMS. And here's the paper that I'm referring to. Okay, so the principle is essentially like this. It's beautifully drawn here in a commentary by Lin and Ermolenko. So this, we have a micromixer, and then a reaction in the sprayer, and then it plunged into liquid ethane, and then we have this product. So when we look at the chip itself, we have a solution, solution 2, they mix together, and then there is a reaction channel of different length. There's either straight or meandering like this, or lots of length. And depending on the length of that channel, we have different times. And then in the end, the reaction products are sprayed onto the grid. And the grid falls into the cryogen. I'm talking about react products because there are several, because we have now a wild mix with everything that is going on, because this is not choreographed, they are not in lockstep. So at any given time, we always have subpopulations mixed together, and classification is an absolute must in order to tease them all apart. Here's the others. At least in the shape it was before. Here's this microfluidic chip, and what you essentially see here are the two inlets, and then here's the sprayer. The sprayer is operated with two additional ancillary gas, and the spraying goes in this direction. Now here you see the grid is mounted on the tweezer, and the tweezer falls down into the cryogen down there, okay? And the whole thing is surrounded by a chamber which controls the temperature and the humidity. There are several applications that were all done in the bacterial translation field, because this is really the specimen that we are looking at. And this has always been the model for the development of all the different methods of cryo-EM. Translation initiation, after mRNA is encountered, the two subunits come together and form a functioning ribosome. Translation termination, after the polypeptide is made, a stop codon is encoded, causing the polypeptide to be released. Or ribosome recycling, after termination, ribosome is split into subunits. Okay, so here you see so many different reactions, and here's the initiation of the reaction. And so we have been dealing with initiation, recycling, and termination so far. And right now, I'm only giving you a few slides about the initiation in order to show the different steps that we're going through. And here's a paper that was published in Nature last year that I'm going to talk about. This is in collaboration with Ruben Gonzalez group. And then there was another paper on translation termination, also published last year in Nature Communications. Okay, so here we go with initiation. And basically, we have the so-called 30S initiation complex, which has the small subunit, a tRNA, the mRNA, and then two initiation factors, IF1 and IF2. And these are the two components that come into the two different channels. And when they come together, they form a pre-70S initiation complex. So everything is together. And then comes a step of GTP hydrolysis, which causes the ejection of one of the initiation factors, IF1, and the reorganization of this heat. And we went into the next stage, which is this 70S initiation. Okay, and then next is PI release. GDP hydrolysis has happened here as a result of this reaction, but everything still stays together. And the PI release is a little long-term process that then results in the release of the IF2. And that gives us, at the times larger than 600 milliseconds, the 70S EC, which is the elongation competent 70S. Okay, so there are a variety of conformational changes that happen on the way to the fully formed 70S ribosome. And the objective was to trap short-lived state in this short-lived state. Now, we were able to have reaction times of 20 milliseconds and one of 80 milliseconds. These are specially fabricated channels for only that are set for that particular time. Now, we found out very quickly that this state cannot be observed. It is already fallen apart, and the only state that we can observe is this. So only one short-lived state can be seen. And here are the materials in the two components. This is what I showed already to you. And here's a picture that shows you droplets. The droplets are spread, but this is now different from the standard cryo-EM in that we don't see the particles everywhere. We only see them in these areas of the droplets. So the data collection is more tedious and involves several grids, and not just grid, in order to get enough quantity, get to the usual resolution in the range of 3 to 4 angstroms. Here's a representative micrograph that shows you essentially it looks rather normal. Then here's a particle picking with rely on 1.2 that shows you what's been picked out. Data collect details. Now, what is really important here is that the treatment of the grid, because we want to have the grid with the right kind of hydrophilicity. Because if that goes wrong, then we get droplets that are too thick. So the ice thickness is a very critical factor here. And then this was done with the Polara, with our 300 kV Polara. Now, I told you earlier that we always observe a wild mixture of everything, and some subpopulations go up in concentration and some go down and so forth. So at each time point, we have several subpopulations intermixed. And so now we can, through classification, we can take them apart, and then we can get high resolution reconstructions of the different components. So this is essentially an inventory of all the reconstruction that we get out from all the time points. And this is the one of the initial components that shows everything that is in the initiation complex of the 30S. We also find another structure which probably comes from some of these here. For the other initial net, which is the 50S, one of the components that enters the input channel. And then I told you there's one intermediate that we cannot see, but we can see at 80 milliseconds the 70S IC, and that still shows the IF2. And then when we wait long enough, which is 600 milliseconds or more, all this is gone, and we get initiation competent that is reorganized by slight rotation of the subunits. Okay, so here is actually by particle counting, we have essentially kinetic data that show us at any given time at 20, 80, 200, 600, how many of these components are there. So here the 50, we start with a lot of 50S, and then 50S goes down. 70S is 0 first, and then it goes up. The elongation competent, and the really interesting one, 70S IC, goes up in concentration and then goes down. Okay, which shows to you that in a few seconds nothing is gone, everything is gone, and you will not see a single thing by standard cryo-EM. Okay. So this is what we found, and now we can look at it in high resolution and exactly see how the components are arranged with each other, and we can make atomic models of these. We also can see conformational changes when we compare the 30S IC with the counterpart on the 70S. So it's a reorganization that takes place here in these components. Okay, so in conclusion, we get several results. We get not only structures, but we can also get the kinetics of the reaction. That's very important. We really can get the kinetic data. It's a beautiful sort of byproduct, and it could even be the main product of cryo-EM. The pre-70S IC could not be captured, as I said. The IF2, IF1 leaves before 20 milliseconds. The 70S IC is seen for the first time, still in the PPI state. The release of IF2 is accompanied by small intersubunit rotation and initiated TNA repositioning in going from the 70S IC to 70S EC. So in general, the general conclusions are short-lived states of any molecular machine can be visualized at a spatial resolution comparable to standard cryo-EM. Now, there are two caveats. One is that we need some more material than we normally have, and we need to optimize this because this is currently a problem. And the other caveat is that the data collection is really spread over a number of grids, so it is more tedious than with the standard cryo-EM. So yeah, I already made this point. Time-resolved cryo-EM can be used to determine kinetics just by particle counting. And further on, we are developing, we are in a process of developing, in collaboration with the engineering department, a chip that might be able to go down to 1 millisecond and below, and which would then be able to follow the switching and gating of ion channels and receptors. And that will be an incredibly a good development. this is not just by downsizing and changing the geometry of what we have. It really uses completely different principles. And that I'd like to show you the various people. Sandeep, who is now in India, Jack Fu, pharma company, I mean, also at the pharma company, when Lee went back to China. So there's essentially nobody left except Prikchat and Chiang Song. These are in my group now, and they are further developing the technology, and they're also actively working on eukaryotic translation project. And Qiang Lin is in the engineering department with his students, and he is developing these very short time channels. Ruben Gonzalez and Kevin Caban are from the chemistry department. They are the specialists in eubacterial initiation, and they also look at the single molecule threat of these molecules. And we have had a long-term collaboration with Howard White, and the driving apparatus that sort of activates everything is also from Howard. Okay, thank you very much.
Thank you very much, Joachim, for this very exciting and stimulating talk. Now you begin to look at not only structure, but the kinetics. It's extremely powerful too. For those audience who like to ask questions, please write your questions in the Q&A. Joachim, the first questions posed is that in bacterial translation initiation slide, you talk about the short-lived channels were designed according to the time between 20 milliseconds and 80 milliseconds. The question is, how were those channels designed?
How are the channels? So they are, these are silicon, they were made from silicon. They were monolithic pieces designed in the nano department of RPI. And we adopted these, the design, but found out that it's a very tedious and expensive process because these waivers need to be done at Cornell. And so we have changed the whole technology to PDMS, which is a plastic, and it's much easier to do. And so these are multiple layers of plastic that are glued together.
Okay. Good.
And so the reaction channel, is simply such a meandering path, and the length determines the reaction time.
Mm-hmm. Good, thank you. Another question is that when we compare structures, many other investigators use other traditional techniques like X-rays or X-FEL, which can give the native conformation at room temperature. What's your opinion in comparing your approach with other solutions scattering approach?
Well, you first talked about X-ray structures, but you don't want to talk about solution scattering, right?
What they...
Solution scattering
You mean the X-ray solution scattering study, not crystallography?
Gets a resolution that even X-ray solution scatter.
Yes.
I'm sorry, we're losing the signal here.
Yeah. The question is, one can possibly. Hello? Yes. Can you hear me? Can you hear me?
Yeah, we got sort of interrupted.
Yeah.
Right. Now it's fine.
Okay.
Yes. But if you mean the, hello?
Yes.
I could not hear you in between.
So the question is, one may possibly use a solution scattering experiment by X-ray, which they can also study the conformations, but at room temperature. The question is, what's your opinion comparing the two techniques, or is there pros and cons in those approaches?
I'm just not aware of solution scattering experiments that can get us into this kind of resolution range.
Yeah.
I wouldn't know how to compare these.
Good. Thank you.
Yeah.
And
but there are other X-ray, experiments and electron-free laser experiments that give us information on conformational changes, but these are really in the femtosecond range and so forth. It's again not comparable at all. The reason why we can do time-resolved experiments is that they are certain short-lived states, but these are really states that are assumed for a reasonably long amount of time. And so they represent sort of kinetic traps, if you will.
Okay, good. Let's try another questions. And when you talk about the kinetic curve, do you concern about the numbers of particles that are included in the final reconstructions? In other words, are you comparing the same statistics, or how many particles do you use?
Well, yeah, this is an excellent question, and I have slides that have something to do with it, but I could not include them because of the time limitation. Because I can only include those particles that I can identify by classification. I cannot include the other ones. Okay, so and when we look at the possibility of doing kinetics, then we have to look at all the different steps in the data processing that goes all the way to the classification result. So the very first question is whether the particle picking doesn't produce bias. Then the next stage is whether the particle collection throws out particles, whether this introduces a bias. So all these different steps really have to be refined and optimized to get really comparable results to the traditional ways how kinetics are done.
Okay, good, thank you. When you prepare the grid, Do you also using a blotting procedures to minimize?
No.
No.
No, not at all. That's the whole idea. And in fact, we can also use this whole sprayer without mixing in order to do grid preparation. It's blot-free grid preparation. It's the whole idea. Because the droplets, they're spread out and then attain the right thickness that we normally get through blotting.
Good.
Okay.
Thank you. So another question raised is, do you need a lot of material to do this kind of experiment? How many?
Yeah. I don't have the number handy right now. It is really multiple times of what one uses for the standard cryo. So we're trying to minimize the amount and to get closer to the normal standard preparation. You can read this. I'm sorry that I have the number not handy here, but you can read it in one of the publications that I showed you.
Good, thank you. So have you tried an experiment that changed the concentration of the reagents? The kinetics also depends on the concentrations of the reactants.
Yeah, but we have not, since this was not the primary purpose, we have not done systematic studies of that.
Okay. Okay. So there are studies that using image processing to define the different mixtures of conformation on the grid. And what's your opinion using other software like Cryodragon?
Like cryo what?
Cryodragon, the software developed at MIT to sort out different conformational states. You use the Reliant. The question is, do you have any opinion using other alternative?
No, I don't. We don't have practical experience with this, so I cannot comment on this. And what I forgot to say is something very important. In whatever classification package you use, you would in experiments you would want to classify all the particles at the same time. You don't classify the individual data collections separately. You classify them all at the same time. And the advantage is that the classification, the scheme is really the same for all particles. And the other very big advantage is that you get always the maximum amount of particles for each of the reconstructions that you have. So if there's a certain subpopulation, I showed you first, where is this? Yeah, here. Right there. See, if you use, these are in gray here, right? So you use all of these and all of these and all of these all together, then you get the maximum amount of particles for a single reconstruction, okay? So you get very highly statistically significant reconstructions. Mm-hmm. That's better than having individual reconstructions that are lower resolution.
Great, thank you. And one last question. are you concerned with some of the particles are defective during the specimen preparation procedure? So the populations of particle you obtain might be somewhat.
Yeah, well, I think this probably needs to be investigated for any molecule separately. For the ribosome, that has been done by the initial developers in at the Wadsworth Center. They they used ribosome and drove the ribosomes through the apparatus and collected them and then performed activity assays, and they behaved completely normal. They made proteins just like before. Okay, so you could see that with this assay you can confirm that you have that the molecule has still functional integrity.
Okay, good. Thank you, Joachim, very much for your excellent presentations and answer only some of the questions being raised by the audience. And we will have some more time that we can spend with Joachim after the next talk. So please stay around after the next talk. And I turn the floor back to Bob.
Bob, you're on mute.
Thank you very much, Wa and Joachim. It's brilliant stuff and many questions raised. So let me pass on to Yi. And of course, at the end, before we go into the broad discussion, we will announce the next symposium, which will be on February 1st. So Yi, could I pass the baton over to you, please?
Hi, everybody. Well, happy New Year. Let me add my greetings to everybody. Joachim, for the very exciting talk. I look forward to bring you back again in the panel discussion. Let me, start by now also mentioning, I'm so impressed by this event. This is the first working day of the new year. We have close to 700 people actually listening just on TEM. I'm just so impressed by the attendance numbers. I look at some of the names, many TEM experts. I also see our program managers logging in, listening to the talks. I see many students, so this is just fantastic. Let me introduce the next speaker, Professor Jennifer Dionne. Jen, it's great honor. Jen is a good friend, a good colleague here at Stanford. He joined the Stanford faculty for about 10 years right now, has been leading a very exciting research group, doing cutting edge research. Her contribution ranging from helping to establish the field of quantum plasmonics. And he also pioneered the methods in combining optical and environmental electron microscopy to image photocatalytic structures. He has been really going to many exciting directions. Her contribution has been recognized by a lot of awards. For example, back in 2011, just a year after he joined the faculty, he was recognized with MIT Technology Review TR35 award. And particularly recent years, some of the very exciting awards she won, including the Moore Inventors Fellow, NIH Director's New Innovator Award. And in 2019, she won the Alan Waterman Award by National Science Foundation. This is only given to usually one person per year across all the fields. In addition to very exciting scientific program Jen established, Jen has been also serving the research community as well. Recently, he stepped up to be the Senior Associate Vice Provost of Research Platforms at Stanford University. Indeed, she's leading the effort to help building up the transmission electron microscopy facility at Stanford right here. She's also serving as a director of DOEs and EFRC, and she's a co-director of Stanford's Atomic Center for Sustainable Energy. She also serves as the associate editor for Nano Letters. With that, let me bring Jen to the podium. It's all yours, Jen.
Awesome. Thank you, Yi, for the amazing introduction. And thanks to Yi and Bob and Hua and the organizers for giving me the opportunity to share my work today. It's such an honor to be here. And yeah, I'm really amazed at the attendance. Happy New Year, everyone. I really hope 2021 brings renewed hope and much health and happiness. You know, I'm here to share with you my lab's work on optically coupled environmental electron microscopy. Oh, I apologize, my laptop kind of got ahead of me. And you might have noticed that my title, The Light Stuff, is inspired by The Right Stuff, like the 1980s drama about the Mercury 7 astronauts and kind of their seat of the pants approach to the space program. And it's in part because I think there are a lot of analogies between a space mission and environmental electron microscopy. So of course, first of all, your sample is going to be heading into vacuum, but you need to find a way to keep it under the appropriate atmosphere, especially if you want to be looking at catalysts and photocatalysts. And then secondly, a lot like a space mission, a lot of the experiments that we're doing in electron microscopy, and in particular in environmental electron microscopy, are extremely sophisticated and complex, and they certainly require a team effort. And in this case, I want to make sure I acknowledge an incredibly talented team of students and postdocs, as well as faculty and staff collaborators who made this work possible, including Bob Sinclair, who helped me you know, lay a very strong foundation for electron microscopy and enabled some of this work. And then finally, just like with a space mission, if you want to be successful, you need to find a way to turn on the lights, you know, especially if you want to probe optical processes. And that's what this talk is going to focus on, how we can bring light to environmental electron microscopy in order to enable atomically optimized or atomically precise photocatalysts and route to a more sustainable circular materials economy. And this is a long-term vision, but in many ways inspired by the fact that if you look to catalysis and chemical manufacturing today, it is an absolutely crucial part of our economy. I think catalysts and catalysis constitute something like 35% of the gross domestic product, or GDP. If you look at the materials that are being manufactured, ammonia is actually one of the most highly produced inorganic chemicals, with over 150 million tons produced each year. And that is such an important precursor, generating enough fertilizer, for example, to feed our world, enabling the production of plastics, of fibers for clothing, and also helping to enable production of pharmaceuticals. And yet production of these inner chemicals is also leading to an environmental crisis, and one that my lab is especially passionate about and trying to devote more and more research into is plastics. And here in the middle of the slide, I just show basically the Great Pacific Garbage Patch. More than 8 million tons of plastic enter the oceans each year, equivalent to dumping a full dump truck full of trash, essentially every minute. So we need to find a way to enable a more circular materials economy, where with the materials we're producing, we find a way to kind of upcycle them and convert them back into their native chemicals. For example, in plastics, could we convert them back into their monomers to make new plastics and enable more efficient recycling, or back into chemical feedstocks like carbon monoxide and hydrogen that can subsequently be recycled to make, for example, sustainable liquid fuels or precursors to other chemicals. And I think there's hope to clean our land and to clean our water with catalysis, especially if you look back to what catalysts and photocatalysts have enabled in air purification. And catalytic converters that use materials like platinum and palladium, of course, have enabled remarkable reductions in smog emissions from cars, significantly lowering the amount of CO emissions and enabling clean air. And here, for example, is just an image of New York City, where the smog in 1973 was absolutely horrendous. 2013, much clearer. I did my graduate work at Caltech, and I remember when I first was thinking about going there back in 2003, people said, oh, you won't even be able to see the mountains in the distance. And of course, now we're fortunate enough to have amazing catalytic converters that have significantly reduced the smog and allow you to see the beautiful mountains of Pasadena. So in any catalytic process, whether to upcycle plastic or enable clean water or clean fuel or clean air, we need to rely on catalysts. And these catalysts are generally going to be nanoparticles that have a high surface area to volume ratio, and they also have sufficient active sites to enable basically faster reaction kinetics as you're transforming one molecule to a different molecule. So as you're transforming the reactants to products. So nanoparticles, are used essentially in every catalytic process because of their high surface area to volume ratio. But of course, when they are operating in kind of a dynamic, reactive, gaseous, or liquid environment, they're going to be undergoing certain transformations. So here, for example, is a paper from Nature Materials published just about 6 or 7 years ago, where you can see that this platinum catalyst is rearranging under oxidation conditions. So we know that catalysts are changing their shape, and of course, that's going to change the number of active sites they have, since usually it's the sharp tips or the low coordinated sites that are the most active. So what my lab wants to do is develop a way to probe catalysts and catalytic activities kind of in situ, in operando, and ultimately we're excited about photocatalytic processes that can hopefully enable a more sustainable, renewable approach to chemical manufacturing. So that's going to require bringing not only environmental conditions to the electron microscope, but also optical excitation. And what we're going to do is take a single particle approach to studying catalysis. In the presentation today, I'm going to just compare a few different types of particles that are different shapes, some of which are single crystalline, including single crystalline metallic cubes and prisms and octahedra. And then we'll also compare that with polycrystalline materials. We'll look at individual particles in this presentation just of one material, namely palladium. But then we're also going to share at the very end some future directions that we're looking at of bimetallic structures, where we can include dopants, in some cases even single atom dopants, where we can study not only the shape and the size, but also how the composition of catalysts at the atomic scale influences their catalytic activity. And in the last 10 years that I've been at Stanford and developed methods within the electron microscope, we've started off with a model reaction that's the hydrogenation of palladium. And the reason for that is because it's not only a very, historical and well-studied model system, especially in bulk, but it turns out that the formation and dissolution of the hydride phase in palladium catalysts and at the surface of these palladium catalysts is often a deciding intermediate step in many catalytic reactions that are hydrogenation based. Oh, I apologize, my PowerPoint seems to have a mind of its own, it's skipping ahead. Here's just one example from Billeter and authors, where they looked at CO2 reduction in palladium catalysts and found that as soon as the particle had hydrogenated, the slope of your production of CO during CO2 reduction was significantly reduced. So the formation of the subsurface hydride phase can have enormous impact on the catalytic activity of palladium-based catalysts. So what happens when you're looking at palladium in a hydrogen-rich environment? Well, the hydrogen first catalytically splits into hydrogen atoms or ions at the surface. And then hydrogen intercalates into the nanoparticle sitting at the interstitial FCC sites. At sufficient hydrogen pressures, you're going to have a phase change. So at low enough pressures, there's not really a lot of hydrogen that's intercalated. Once you've gotten to high enough pressures, you're going to have a phase change from the alpha phase to the beta phase. That phase change is accompanied by a lattice expansion. And I'll mention that this not only occurs in bulk, but can also occur at the surface. And I'll share with you how we can probe kind of that subsurface formation. That phase change is associated by about a 10% lattice expansion, or about a 3% change in lattice constant, 10% lattice expansion in volume. And that's because the hydrogen is a misfitting sphere in those FCC sites. So there's quite a bit of strain induced by having the hydrogen intercalate. And like I said, we're going to be using environmental and optically coupled transmission electron microscopy. We synthesize our nanoparticles using colloidal syntheses, in some cases just conventional syntheses, in other cases new syntheses where we can get almost atomically precise control over the catalyst. And then we're going to use our electron beam and a combination of electron spectroscopy, for example, electron energy loss spectroscopy, to probe kind of the electronic and optical properties of the material. And we also use diffraction to probe the structural properties. And we can do that both with and without illumination. And I'll focus on the first part of my presentation just on the results without illumination so we can understand what's happening in the dark. And then as we introduce light, we can see how light, and in particular nanoscale control of light and excitation of surface plasmons, can change the reactivity and reaction intermediates. So how is it that we're probing hydride formation? I mentioned that there's a change in lattice constant. So as we go from the hydrogen poor to the hydrogen rich phase, there's a 3.5% change in lattice constant. So that's obviously something that we can follow using conventional diffraction. And then similarly with electron energy loss spectroscopy, that's probing kind of the band structure or the material's electronic properties. It turns out that when there's hydrogen in the lattice, there's actually a 2 electron volt resonance shift of the bulk plasmon resonance. And here I'm showing you the electron energy loss spectra, whereas you go from the alpha phase, you have a peak right around 8 electron volts corresponding to the bulk plasmon resonance when you excite the nanoparticle in the center. And then as the particle gets fully hydrogenated, there's an enormous shift in that bulk plasmon resonance to less than 6 electron volts. So that gives us a huge signal to noise ratio with which to follow where the hydrogen is intercalating into the lattice. And I'll mention that in my presentation, we're mostly going to focus on where hydrogen is going in, but my lab has recently developed techniques to follow also which products are going out of the particle. Okay, so how do shape and crystallinity affect the thermodynamics of hydrogen intercalation? So first, just in the dark, what we're going to do is put our nanoparticles in the TEM, put hydrogen in at different pressures, and then just follow the equilibrium isotherm. So we change the hydrogen pressure, waiting in each case about 30 minutes to make sure the system has equilibrated. It depends obviously on the amount of time for equilibration on the particle size and shape, but generally after about 1 to 2 minutes, most particles have equilibrated. So you'll see here for different size single crystalline nanocubes and single crystalline prisms that the isotherms show very abrupt transitions between the alpha phase, represented over here by essentially a 0 or close to 0 fractional percent of the beta phase, and the fully hydrogenated phase. So we have these extremely sharp transitions for the single crystalline nanoparticles. If you compare that to the polycrystalline nanoparticles, for example, the icosahedral nanoparticles, you'll see that the isotherms are considerably more sloped and gradual. Let's dive into a little bit more detail about what locally the hydrogenation state is of each nanoparticle. And if we look at the single crystalline nanoparticles like the nanocubes or the nanoprisms, and we focus our beam down and then raster scan it over the nanoparticle, we can create maps of the system in equilibrium. And I'll mention this is actually where cryo comes in, because these are longer scans. So what we have to do is flash freeze our sample to below or at 77 Kelvin. So that way the hydrogen essentially remains trapped within the nanoparticle. So we're in equilibrium, we flash freeze it, so that way we can construct essentially EELS maps of what the phase is. And if you look at the spectra, for example, when the electron probe is focused at this green point in the center of the nanoparticle versus at the pink point, you'll notice at low hydrogen pressures, we always have a peak right around 8 electron volts, whereas at higher hydrogen pressures, we always have a peak just below 6 electron volts. And when you map that over the entire nanoparticle, we find that for single crystalline particles, we're always in a single phase regardless of hydrogen pressure we're at. So at low hydrogen pressures, we're always in the alpha phase. At higher hydrogen pressures, we're always in the beta phase. That is in contrast to the icosahedra, these polycrystalline nanoparticles, where there are certain hydrogen pressures where you find a multitude of phases in equilibrium. So the icosahedra are actually compressively strained. So there's more strain at the core of the particle than there is at the surface. And that strain makes it much harder for the hydrogen to intercalate into the core of the icosahedra. So at intermediate hydrogen pressures, we find that the core is unhydrogenated, and it's really only the outermost shell that is hydrogenated. We can also look at individual crystallites within the icosahedra, and here, just in the interest of time, I'm skipping over how we do the experiment. But we have essentially tetrahedra crystallites that comprise the icosahedra, and we can look at diffraction to identify how each tetrahedral component of the icosahedra has transformed in equilibrium conditions. And we wind up seeing that essentially the grain boundaries decouple the crystallites, leading to what we've called mosaic loading. So when one tetrahedral crystallite transforms to a hydrogenated phase, that basically applies strain to the neighboring tetrahedra, making it a little bit harder for the neighboring ones to transform. So you have to get up to much higher pressures to enable complete transformation to the beta phase. So everything I've discussed so far is in equilibrium. How can we image the reaction kinetics? Well, there we can rely on STEM and EELS contrast. And here, when hydrogen has been introduced into the chamber, you can see that the contrast of this individual nanocube has changed. If we look just at some time snapshots of that moving, we can actually identify that contrast as where hydrogen is intercalating by combining not only the STEM contrast with diffraction and local EELS studies. So we see that the beta phase actually starts from the cubes of the corners, and then it appears as if some of the surfaces kind of get wet with hydrogen. So we have this subsurface loading, or what appears to be subsurface loading. The beta phase eventually stabilizes along one of the 100 fronts, which is one of the lower energy configurations for this alpha beta phase coexistence when you're not in equilibrium. That hydrogenated phase then continues to move with this straight or flat phase front across the nanoparticle until eventually the alpha phase is pushed out again through another corner of the cube. We wanted to know if those low resolution STEM images that indicated there was surface wetting or surface hydrogenation actually occurred when we could get atomic scale resolution in an environmental condition. And recently, thanks to advances in direct electron detection and Gatan's K3 camera, we're able to do environmental imaging at atomic scale resolution. Here I'm showing you an example of one of those single crystalline prisms where you can see that, again, the hydrogen enters from the tips, but there is actually this subsurface of hydrogen. And if you look at the lattice fringes along each of the corners here, I'm just showing you one of them, but we actually wind up having confirmatively subsurface beta phase within the first few monolayers around the entire prism. So I mentioned that we have intercalation beginning at the corners, subsurface wetting. Eventually, the hydrogen-rich phase stabilizes along a particular direction, so that way it can propagate along the nanoparticle. And when we have alpha-beta phase coexistence, we're going to have quite a bit of strain within the nanoparticle due to that 3 or 3.5% lattice mismatch between the alpha and beta phases. And during the transformation, we can look at diffraction of individual nanoparticles. So here I'm showing you diffraction of an individual nanocube. We're zooming into one diffraction point, and you can see right at the beginning of the transformation, that diffraction point is actually 2 subpoints corresponding to the alpha and beta phase, depending upon whether you're looking at the inner or the outer ring. And you can see that the full width half max of this diffraction point is quite tight. So the particle is still a single crystal. As the transformation progresses, you can see that individual diffraction point actually broadens a bit, implying that various lattice imperfections are forming. Those could be various defects. They also could be just quite a bit of strain, coherent strain within the nanoparticle. But then amazingly, at the end of the transformation, once the hydrogen has moved across the entire nanoparticle, the full width half max of the diffraction point tightens up again. So it becomes less broad. In some cases, it actually becomes smaller than the original particle. So this shows that the nanoparticles, at least below a certain size, have this ability to self-heal. So even though there's strain involved in this transformation, the particles can go back to being an extremely good single crystal at the end of the transformation. Okay, so I wanted to share with you that it's not only the single crystalline nanoparticles that have corner loading. We can also look at octahedra. The reason we looked at octahedra is because, well, those cubes were, 100 terminated. Was it like the surface termination, or was it the fact that the corners were like 111 terminated? So we want to choose a single crystalline nanoparticle that had 111 surface terminations. Turns out that's less relevant than the low coordination number at the sharp corners. So it's really the corners that determine the corner loading. And you can get that also in polycrystalline nanoparticles, for example, nanorods, where again, the hydrogen intercalation is always going to begin from those sharp corners. And this is what brings me to light, and in particular, how we can use light not only to change the reaction kinetics, but also to change where the reaction is occurring. So we worked closely with Gatan, starting I think about 5 years ago, to develop methods to introduce light into and out of the environmental electron microscope. We essentially surround our sample with a parabolic mirror. The mirrors have holes through it to allow the electron beam to pass through, and actually it's a slit, so that way we can also do tomography. And we designed the system to enable not only high optical incoupling efficiency, but also high optical outcoupling efficiency. And when we're looking at light, we wanted to start off with our palladium nanoparticles, which are not particularly good light absorbers. So what we did is couple those palladium nanoparticles to a gold nano antenna. And this is an antenna reactor design inspired by the Hollis and Norlander labs, and in particular by then a graduate student, now my postdoctoral student, Dane Swear, who first investigated how you can kind of couple an optically active material like gold with a catalytically active material like palladium to enable more efficient catalysis. So we have a gold disc that we fabricated by electron beam lithography, and then we sprinkle on the palladium nanoparticles and the catalyst. And for this particular antenna reactor pair, we can again look at the equilibrium thermodynamics. What we're going to do is look at dehydrogenation. So we'll set the pressure just above the unloading pressure, and without any illumination, the system will stay in equilibrium. So we optimize the electron beam conditions such that it does not perturb the system from equilibrium within a reasonable time frame for the experiment. It's only the light that's going to be pushing the system out of equilibrium. And here's an image of the diffraction pattern. We've turned the light onto that antenna reactor pair. You can see for a while nothing happens, but then after a few seconds, the diffraction pattern actually expanded, corresponding to the transition from the beta phase to the alpha phase. So what we've learned is that there are actually 2 time scales that govern the transformation. We call these time scales, the induction time and the reaction time. So if you look at the diffraction pattern and the number of phases as a function of time, like I said, we turn the laser on initially, there's a time period across which nothing happens, and we call that the induction time. And then suddenly we wind up seeing the system transform from the beta phase to alpha and beta phase coexistence. So the system is in a state with phase coexistence, and then ultimately the system transforms to purely the alpha phase. So the time between when we wind up having coexistence and when the transformation is complete is something that we call the reaction time. So how do these two timescales vary upon illumination? So now we send in different wavelengths, and we see that for wavelengths that are, for this particular sample, roughly in the green and blue and also in the near infrared, the induction time lasts a long time. So essentially nothing really is happening upon illumination when we're at very blue and very red wavelengths. But when we're here in this intermediate regime at wavelengths of, say, 630 nanometers and 690 nanometers, we find that the induction time is extremely short, so less than 10 seconds. And here I'll just mention that we had passed our light source through a bandpass filter, and the length span of each of these bars corresponds to the bandwidth of the illumination. So each of these dips where we have significantly increased kinetics, in some cases up to 10x increased kinetics, correspond to where we have plasmon resonances. So if you look at the EEL spectra of this mode, we wind up seeing two peaks right around 630 and about 720 nanometers that correspond to the plasmon resonances. What was super interesting about this is the lower energy mode actually corresponds to a dipolar plasmon type resonance. It's optically excitable. You'll also see this mode in cathodoluminescence. But this higher energy mode would normally be dark, meaning you normally wouldn't be able to excite it with light, but by virtue of the substrate, it becomes optically excitable. And I think this is pretty exciting because the catalysis community is focused, for many, many decades on how the substrate can influence activity. That substrate not only influences, say, the strain that a particle might feel when it's sitting on the sample, but it also changes the local density of electronic states and the local density of optical states, such that you can wind up getting some of these plasmonic modes to be optically excitable just based on the substrate. Okay, so we have 10x increased induction times. What about the reaction times? You can see that the reaction times also kind of show some dips corresponding with the plasmon resonances, but by and large, they're wavelength independent. And that's because the induction time influences how the hydrogen is catalytically splitting at the surface, whereas the reaction time is really going to be determined by how the hydrogen is diffusing within the nanoparticle. And then, of course, since we have the ability to visualize in situ how hydrogen is intercalating, we can see how upon illumination, and in particular how excitation of plasmonic hotspots that are concentrated at the gap between the antenna and the reactor influence site selectivity. So in the dark, when you look across more than 25 antenna reactor pairs, there's an arbitrary corner at which you're going to have the nucleation of that alpha phase. But once you turn the light on, and when you're on the plasmon resonance, more than 75% of those antenna reactor pairs load near the electromagnetic hotspot. So namely near the tip that's closest to the antenna. And those that don't load near the tip do so mostly because the separation between the antenna and the reactor is too far. One thing we also notice is that upon illumination, you not only have nucleation of the new phase when you're closest to the antenna, namely the gold disk, but we also seem to see this edge nucleation. And I mentioned that we always saw corner nucleation followed by surface wetting, but upon illumination, in certain cases, we saw what seemed to be this edge as opposed to corner nucleation. We didn't know if that was mainly just expedited kinetics, like for example, maybe we didn't have the time resolution to capture the very rapid corner loading, or actually a new mechanism. So to explore that, what we started to investigate are these crossbar systems. And in particular, what we wanted to know is whether excitation of surface plasmons could activate a non-active catalytic site. So you'll mention or recall that these bar-shaped structures or rod-shaped structures also loaded from the corners, these nanobars are also really exciting catalysts, but a lot of the surface area is essentially unused. The bar is, I think, a pretty extreme example, but even if you look at a nanoparticle, the catalytic activity is highest at the corners, but a lot of the faces are essentially being unused. Can we come up with a way to make these non-active sites, active, and in some cases even the preferred site, so that way we can enable higher turnover frequencies, higher product efficiency, and in some cases even product selectivity. So like I said, we're going to explore these palladium nanorods. What we do is put them on a gold surface, with the idea being that when you have a gold surface or a gold bar underneath, you have an electromagnetic hotspot that now corresponds with one of the faces of your palladium bar. So you might be able to induce hydrogenation at that face or at that non-reactive site. So again, we use electron beam lithography to create this crossbar geometry. We can use cathodoluminescence and electron energy loss spectroscopy to confirm that the electromagnetic hotspots are at the junction between the palladium nanobar and the gold bar that's underneath it. You can also see here when we fabricate that gold grating that the resonance is mostly in the red. And then what we do is use dark field imaging and electron microscopy to isolate individual crystallites within these nanobars. So that way we can follow the hydrogenation and dehydrogenation of single crystallites. So when we're in the beta phase, the crystallite that we're going to be looking at is the one that's actually going to be, as you can see here, closest to the gold bar. So that crystallite will be lit up or have bright contrast when we're in the beta phase, and it will appear dark when we're in the alpha phase. So here's just one image, and recall that in the dark, we only had loading and unloading from the tips. But what you'll see, and I think it starts right here around the 14th second, is that upon illumination, we can actually get hydrogenation and dehydrogenation from the center of the bar. So I'll play that again just so you can watch the dehydrogenation, in this case occurring from the center of the bar and then moving across one side and then over to the other. And then once we turn the light off and we increase the pressure, we can get the system to hydrogenate again. You'll get to see the contrast in just a few seconds, where now you can see the hydrogenation is again occurring from the tips of the nanoparticle. So I think this is extremely exciting because here, by virtue of exciting localized surface plasmon resonances, we can actually make more of our catalyst surface catalytically active and basically enable what otherwise would have been kinetically unfavorable transformations. And this paper is in press. It should be out in about a week. Okay, so what's next? Oops, hopefully I can get my PowerPoint to go. One of which is creating atomically precise catalysts. So now that we've established a baseline for how size and shape and crystallinity all influence, hydrogenation of the bulk of the nanoparticle, as well as the surface, and how light influences that transformation, we're starting to create bimetallic catalysts. So instead of relying on those antenna reactor systems, now we put the light absorber directly in the material. And in fact, many of these bimetallics are very exciting catalysts and photocatalysts. So we can see how the placement of individual atoms in these bimetallics and how the composition influences, subsurface hydrogenation. We can also look at photocatalysis to see, for example, how single atom dopants and catalysts influences their photocatalytic activity. And then we can also move beyond gaseous environments and start to look at liquid environments. And here, for example, are some first experiments where we looked at electron energy loss spectroscopy in a liquid cell, and we could confirm that we had an aqueous environment surrounding our nanocatalysts. And then just as a second future direction, I'll mention beyond catalysis and sustainability, I think there are some extremely exciting opportunities in quantum information. And I recently was fortunate enough to be part of the DOE QNext Center that's based out of SLAC and University of Chicago with Joanne Hewlett and David Auschalam as the principal investigators. And because optically coupled environmental electron microscopy gives us atomic scale resolution, we can start to look, for example, at silicon vacancy centers and nanoparticles and nanodiamonds, and also at various quantum optical defects in two-dimensional materials, and begin to understand how the atomic scale structure influences quantum optical emission and quantum coherence, and also use simultaneously the electron beam in various gaseous environments to deterministically place emitters. So I think this is an extremely exciting direction. So I'll summarize, by saying that I think environmental electron microscopy offers, the right stuff and the light stuff for sustainability and for a number of other exciting applications. What I'm most enthusiastic about is hopefully trying to work on advances in optically coupled ETEM that can enable a circular materials economy with new atomically precise photocatalysts that are engineered for not only high efficiency catalysis, but also product specific reactions. So I shared with you how we can enable optical excitation within an environmental TEM for dynamic imaging of reactions at the atomic scale, and now with atomic scale resolution, thanks to direct electron detection. And then I also shared with you what we've learned about nanoparticle phase transformations and light-driven phase transformations. These single crystalline nanoparticles maximize energy storage, so they only exist in one phase in thermodynamic equilibrium compared to the polycrystalline nanoparticles. And then light enables significantly increased reaction rates, as well as catalytic site selectivity and the opportunity for and favorable intermediate states. And what we're excited, in addition to those two future directions I mentioned, is linking these single particle properties to reactor performance experiments that we're doing now in collaboration with Matteo Carnago. So I'd like to thank all of my students and postdocs who made this work possible. I'd also like to thank our funders, especially the Department of Energy and recently the NSF through their Waterman Award. And thanks to all of you for your attention. I am happy to take any questions.
Thank you very much, Jen, for the very exciting work and also great talks. Yeah. So there are a number of questions now flowing in. For audience, please type in your question in the Q&A. But Jen, let me start by asking you the first question. Looking at your beautiful video of, alpha phase become beta phase, the snapshot of that. If you look at the same particle, put hydrogen in, alpha become beta, and then you pump hydrogen out, beta come back to alpha, tracing the same particle, and then would that boundary between alpha and beta is kind of reversible, follow the same boundary or different boundary?
Yeah. No, that's super exciting. Yeah, I didn't go into the cycling data here, but it's true. When you look at the same particle systematically, you'll get loading generally at the same corner, and you'll wind up tracing out the same phase. So initially you have kind of this diagonal phase front. It looks like it's along in the cube, the 1, 1, 1 corner, and then it stabilizes along the 1, 0, 0 interface. So generally it seems like we're getting loading at the same corner. Unloading appears to be a little bit more stochastic, but it's based on limited statistics, so it's hard to know why that's the case. And I think that's a really interesting question. Like, as we start to build up, looking at hundreds of nanoparticles in multiple cycles, to what extent is it like the particular coordination of a certain nanoparticle that's governing that loading, and what is it about that? And especially is it the atomic composition, the configuration? I think that's really important for catalysis.
Sounds really interesting. If you do multiple cycle, if we all nucleate from the same spot, that might be the certain defect right there, or certain composition right there.
Yeah, exactly. And that, is actually really important, especially as we wanted to demonstrate kind of new mechanisms for kind of nucleation with plasmon excitation or with optical excitation, that's where we need to do more statistics over the rods to always show, okay, it's always loading from, you know, the tips. Generally, once it's loaded from one tip, it seems to have a preference for that tip, but then upon illumination, you can kind of reversibly go between, you know, face or edge nucleation and tip nucleation, depending upon whether the light is on or off.
Yeah. So the second question this is from audience. I think Bob Sinclair probably would like to ask the same question as well. So every time with this study with dynamic process right there with beam, I think this is a question from the audience asking, what's the effect of the beam to the hydrogen? Any insight on that? Would you cause the hydrogen to escape under the beam?
Yeah, absolutely. And that's why, you know, a number of our experiments we do with, you know, TEM, essentially the lower the electron dose. I mentioned that when we start doing STEM, so when we focus the beam down to about a nanometer spot size in order to do electron energy loss spectroscopy, there obviously you have a much higher local dose, and that would cause dehydrogenation. So we've done studies allowing us to investigate, you know, at what doses you start dehydrogenating the nanoparticle, you know, how much time does it take to dehydrogenate it when you focus the particle? So to create those EELS maps, like if you're doing more than just a millisecond acquisition at a time and you want to, you know, do like a more thorough analysis over the entire spatial area of the particle in STEM, you need to rely on those kind of flash freezing approaches where you essentially kinetically trap the nanoparticle by like plunging it into liquid nitrogen while doing the in situ measurement. So we essentially quickly cool down our sample so that way the hydrogen remains trapped there, because otherwise the beam is going to cause dehydrogenation when you're in scanning mode.
Yeah. One more question. This audience is asking, looks like it when you do the hydrogen reaction, it's one of the corners or apex to form beta phase. Did you see several of them at the same time? For example, for the cube, do you see kind of 3 corners having beta phase all going?
Oh man, that's a super fascinating question. It doesn't happen very often, but obviously it can, right? Like nucleation is a random process. So, you know, generally you're going to get it occurring at one of the, you know, 8 corners. We can only see 4 in projection. But there are times where we've seen multiple corners nucleating simultaneously. And that can give rise to really interesting, you know, striped phases within the nanoparticle. So we collaborated on one of our papers with Martin Bazant, and he was able to kind of model this striped phase. It really depends actually on the size of the nanoparticle, because like I said, there's strain with that alpha beta interface. So the larger your nanoparticle, the easier it is to nucleate at multiple corners or multiple tips and basically have that, you know, set of multiple phases, 2 phase fronts moving across your nanoparticle. Once you're below a certain size, and I'd say that's of order, you know, 100 nanometers for an edge length or a rod length, you usually just get one corner winning out, because otherwise the energetic barrier to have multiple interfaces is too high. But when your particle's large enough, you can kind of decouple those interfaces and get multiple loading events.
Yeah, this is great. This is really, really interesting. So this really made me to think about the size effect, right? The particle you have, for example, the cube or triangle shape, they are like what, 100 nanometers, something like that.
So we've, yeah, in this presentation, I kept it simple, but we've gone all the way from about 10 nanometers up to 300 or 400 nanometers.
Yeah.
And yeah, basically it's below a certain size regime, and that's kind of the size I focused on in this talk, that you wind up seeing kind of these coherent, you know, abrupt phase transformations, you know, one corner nucleating, and then the ability to get a lot of the kind of selective plasmonic control.
Yeah, that's great. And there's one more question. Let me ask before. After that, I probably will bring back Joachim to the podium. About, let's see, can in situ liquid yields measure metal plasmon peaks even though the low energy range of the plasmon would be overwhelmed by the inelastic yield signal from liquid?
Oh, absolutely. That's the data I was showing on the last slide. And I stopped screen sharing, but I think it's recorded so the speaker can go back and look at it. We're writing up that paper now. I think it's, just a combination of technique development, but I don't want to, underestimate, luck playing a role. You have to have, the right thickness of water so that way you're not being overwhelmed by the inelastic scattering there. But it's super exciting. I think we've now developed liquid cells where we can have, just enough water to surround the catalyst and the right beam conditions so that way you can do low energy plasmon yields and actually measure those plasmonic particle resonances and confirm that indeed the particle is in water based on those yields peaks. So I think it's pretty exciting. And what we're doing now is flowing in different inorganic solvents so that way we can turn the plasmon resonances shifting as the effective refractive index is, as you might expect.
Okay, Jen, thank you so much for the great talk. Now let me bring also Joachim to the podium. Joachim, if you want to unmute yourself, please do so. And Bob, certainly feel free to also chime in to have a panel discussion. Let me start by asking one question, maybe back to Joachim first. I was really fascinated by the Timeresolved cryo. And this is something we really want to do, to use a Timeresolved cryo for material science as well. One question I have is, I saw your beautiful, method of microfluidics mixing and using the channel length to determine the time, right? This is kind of time resolution you're trying to get. And if I ask a question and say, well, would you be able to, what will be the pathway to get to millisecond time resolution? Well, you are showing that, tens of milliseconds, hundreds of milliseconds. If you want to go, more or less 1 order of magnitude, shorter time, anything in your mind you think this could be achievable?
Well, this is all right now. There are feasibility studies, and every single step that we use really needs to be sort of redesigned and reimagined. And one is one very crucial one is that one cannot afford to do anything like plunging. This is it's too time consuming. And so there's something that already Peter Peters did, which is to use a jet stream of the cryogen hitting the grid just at the moment when the material arrives, or a very small time interval afterwards. So this is all. Everything needs to be redesigned. There's no plunging that can be done in these terms. And the reaction time, controlling the reaction time is not such a big problem. The mixing can be controlled and can be further reduced in time. So the main challenge is really to make this cryo transformation possible without plunging.
Yeah. So I have one idea jumping out. I think with the team here, we kind of discussed that a little bit. Right now, looking at Jan's capability of optical excitation. would this idea be possible if you can already freeze in your sample and already freeze right there, and then somehow using the light to kind of locally heat it up to a temperature, because the light can get to femtosecond, picosecond fast, right? Heat it up, and with some certain duration, and then you remove it, and then you quench back again to improve the time resolution. I'm just throwing out an idea and see what Jen, what do you think about this the combination of optical part with Joachim's the cryo part to study the dynamics.
Oh my gosh, I think it's super exciting. And I'll just mention, I think Hua and W.E. Morner have done some really exciting studies kind of combining super resolution optical microscopy with cryo-EM. We're essentially trying to combine kind of 2-in-1 instrument with the optical excitation, but I think there's a really cool technique to look at light-driven processes by basically, combining optical excitation with Joachim's approach to then kind of freeze it and then try to, capture light-driven motion that way.
And also, by the way, I think our Nobel Prize winner W.E. Morner is in the audience.
Okay. Hi.
Raphael, can you check whether W.E. is still there, and maybe bring and see W.E. is available to come to the panelists?
Yeah. I'm not familiar with the collaboration with Hua. Have you published this anywhere, Hua?
No, we haven't published anywhere yet, but a lot of works are being done here locally in integrating different methods. W.E. is now showing up, so W.E. may want to comment. W.E.?
Yeah, sure. Well, let me clarify one thing. What we've done with light and cryo-EM with Hua, cryo-ET with Hua, is to look at single molecules and use them to locate and find positions of specific proteins in the cryo-ET image. And this has been published. This is in PNAS. And so this is a way to make sure you know which objects in the cryo-ET have a particular identity that is determined by the fluorescence. This other issue of using light to trigger reactions, we're not doing yet. And that is something that, of course, has been done for a long time in different other contexts to, for example, take a crystal and stimulate a change in the proteins and then do quickly an X-ray. And so that kind of an idea is a great idea that could be applied to the cryo-EM situation perhaps as well.
Yeah, true. Absolutely.
So, and Hua and Bob, certainly feel free to chime in to ask questions. So I also see exciting opportunity as well to now combining cryo. We just discussed about using light, and potentially there could be more electrical signal. How do we bring it in? And also the magnetic signal type of freedom control, that would be fantastic. I don't know whether Jen and Joachim, you have some idea, and also W.E. as well, and any thought you want to discuss with the audience?
Yeah, I don't. I offhand, I really don't know an application, but I can imagine that there are many biological processes that involve that are light sensitive, and so they could be controlled in this way, and we can well see how these technologies could interlace in these
yeah, I mean, biology is a little outside of my wheelhouse, but like Joachim mentioned, and I think I mentioned W.E. because I just thought the work that he presented on and that was published in PNAS was so beautiful. Yeah, I think there's a lot of opportunity to look at light-driven processes, for example, in bacteria, but then also use optical emission. So, develop electron stable fluorophores where you can locally excite that fluorophore. And that's where kind of some of the super resolution ideas come in. I think bridging what WE and WA have done and maybe what, my lab could contribute, developing electron stable fluorophores where you excite them with the electron beam, but then can look at the luminescence in the TEM. And then you mentioned the idea of, adding in various biasing, right? Like electrical biasing and magnetic biasing. Maybe Bob just speak about magnetic biasing, but electrical biasing, super exciting, especially in the quantum realm. Something that we had put into the QNext proposal is looking, for example, at optically coupled spin qubits and how when you're electrically biasing them, you can change the quantum optical emission and kind of probing that in the TEM and looking at how like atomic rearrangements are influencing spin coherence, I think is a really cool opportunity. Of course, whenever you're doing, multimodal, biasing, these are extremely complex experiments. So each time you introduce a new knob into the TEM, we always worry about beam effects, but then as a question came up, you also have to worry about, well, what is the influence of the light? What is the influence of the electron beam? And what is the influence of the electrical bias? So you need to make sure as you're introducing one new knob each time that you're, making sure you're doing all of the controls, and the likelihood of failure each time you introduce something new into the TEM obviously increases. I don't know how, perhaps exponentially, but I think it's a really exciting opportunity. Like I view electron microscopy and especially in situ EM now as just this enormous playground for exploring, atomic science and being able to create, atomically precise materials and kind of understand how sophisticated, machines like cells and proteins are behaving.
Well, you have the luxury that you can do these experiments in the TEM directly without cryo. As soon as we have the molecules or any material inside of a water, of an ice layer, then it becomes much more difficult to do these experiments directly in the EM. So I'm not sure whether anybody has attempted to do anything like this for biological specimens.
I just want to point out David DeRose in the audience. He points out that some years ago, both Bob Glaser and Richard Henderson have to use the light to trigger the reaction on bacterial adoptions. And I believe that has been published. In fact, they have to do it in the dark room
Okay.
in experiments. Yeah.
Well, dark room shouldn't be a problem, right?
In those times, I believe the specimen was probably embedded in glucose rather than in ice. But I think with the modern apparatus, it probably can be implemented in ice. In different types of specimens as well. Yeah,
that's interesting. Yeah, I'm wondering when was this done and what publication?
Richard, I don't know. Richard is still or Bob still on the lines or not. Maybe we can unmute them. They make some comments.
Or you could just send the an email around.
Right.
Yeah.
So. Well, very exciting discussion right here. I think our time is getting close. Bob, I will hand this back to you. Do you want to announce the next event again?
Yeah, well, thank you very much, both of you, for just fantastic talks, and for Yi and Hua for leading the discussions here. And so glad there's been so many questions, which we can't get to answer them all, but I believe that they can be transmitted to the speakers for their written replies rather than the verbal ones. Yeah, I think this is a fantastic start to the new year. And as I said, we will be convening again on the first Monday of the month, February the 1st. And we have two former MSA presidents who will be speaking next time. Bob Glaser, who is Wachu's advisor from the past, and David Smith, whom many people know from Arizona State University. And so they'll be sharing their titles and soon with us. And we should expect to have another very exciting session. But I'd like to thank once again all the people who've contributed to the discussion, and especially our speakers for getting us off to a great start for 2021. Thank you.
Bye-bye.
And to WE as well.
Thank you.
By our, unnoticed invitation to join in.
Thank you.
Thank you, everyone.
Yeah. Happy New Year. Bye now.
Bye.